type 4 and moving into some of the transplant rejection issues that we were alluding to. So what's going on here when we have a donor, in this example it's a kidney. So we have two different pathways. We have a direct pathway and we have an indirect pathway. And there's this incompatibility between the graft and the host where they're recognizing typically antigens that might even be part of the MHC class. Likely it's part of the MHC class. Remember, this major histocompatibility complex is, you know, your individual cells label telling your immune system that it belongs to you. And when it doesn't see that, it identifies that it's supposed to do something with it. And so it's based off of the MHC classification. And we have a situation over here with the direct pathway where the antigen presenting cell is found in the graft. And the graft, in this case, is the donor kidney. So the donor kidney has an antigen presenting cell that's within that tissue, and it's showcasing this class 1 MHC molecule. And you trigger this downstream event causing activation of CD8 cytotoxic T lymphocytes, and those cytotoxic T lymphocytes are going to then find the donor or the graft renal tubule and start destroying it. Simultaneously, you have MHC class 2 seeing the helper T cell, and you're going to start triggering an inflammatory response against this renal tubule. So this is the direct pathway. The indirect pathway is very similar, but the difference is the antigen presenting cell is in the recipient. So the person receiving the kidney, the antigen presenting cell identifies in the graft for an antigen, phagocytoses it, displays it, and triggers activation of macrophages via more of an indirect pathway, because the step is it has to find the antigen first. But the whole point here is you're identifying MHC class as being foreign, you display that, and you trigger two pathways. You involve T cells, both CD4 positive as well as CD8 positive. Okay? Now, there's an effective treatment or an effective blocker, if you will, or mitigator, which is called an immunosuppressant drug. Now, this immunosuppressant drug is rather effective, but not bulletproof, let's say. And the reason it's effective but not bulletproof is it really targets this direct pathway with CD8. So you can recall back to this figure from last unit where our CD8 cytotoxic T lymphocyte is naive and it's seeing the foreign antigen. In this case, it's an MHC class antigen. And in order to stimulate effector populations and then memory populations, there's this co-stimulation that takes place by the release of interleukin-2. You guys remember this slide? Now, what cyclosporin does in the family of immunosuppressant drugs, like cyclosporin, it blocks the release of IL-2. So you never get this co-stimulation, and you never get activation of that naive CD8 T cell. So the direct pathway is no longer functional, right? This direct pathway no longer is in operation. You still have the ability for some indirect problems via inflammation. And so you may actually put the patient on not only immunosuppressant drugs, but you may put them on what? A low dose of anti-inflammatories, corticosteroid treatment long term. So that way you're trying to mitigate the main pathway of the direct pathway, as well as the side pathway of the indirect pathway, which is an inflammatory mediated event. Okay, so it's not uncommon to have cyclosporin plus a long-term anti-inflammatory like corticosteroid on board with our transplant patients. Well, if we don't do that, what happens? What does it look like? Well, here's a picture, again, of a kidney. And we've got histology showing the progression of what happens with this type 4 hypersensitivity. So on the bottom left, we've got hyperacute rejection taking place. You've got early neutrophil infiltration on that bottom left. In the next panel, just to the right of it, in B, you've got additional inflammatory cells coming in. In C, the third one over from the right, now we start to see inflammatory cells proliferating within the intima of the vascular elements, and you're starting to get smooth muscle cell thickening is taking place. And then over here on the far right, in panel D, you've got arterial sclerosis. The lumen of the vasculature is actually replaced with smooth muscle cells. And so this would be an example of vasculitis within the glomerulus. Okay? So, rejection that takes place is our secondary response after we become sensitized. You develop these antibodies to the MHC class that's foreign. You circulate those around, causing a type 4 hypersensitivity utilizing CD4 helper T cells. You also, through the direct pathway, trigger cytolysis, which is this very specific targeted apoptotic mechanism using CD8 positive T cells. And you block off the vasculature in this kidney allograft. Allograft meaning from the same species, allo meaning same species. And ultimately, if you block off, here's the lumen. You see this opening right here? That used to be all the way over this direction. And the blood vessels start closing off. You start with inflammation of the glomerulus, more inflammation. Now you start with thickening of the vascular wall within the glomeruli. And over on the vascular element, you start blocking off blood flow. If you block off blood flow completely, then you damage the tissue ultimately. All right. So this goes back to the question that was appropriately asked here in the back. What is the pathogenesis of autoimmunity? And maybe it's more appropriate to put the slide in the beginning of the lecture for your question. Okay, if you're struggling with it, that tells me that maybe it's a little out of sequence. Okay, but so that was a very, very appropriate question. Usually by now in the lecture, so maybe you're just advanced, usually by now in the lecture, students are asking, okay, I get this, but how does it all fit in? The idea is this breakdown of self-tolerance mechanisms. This is the leaky part that we were talking about. It's not supposed to do this because we screen for them. We actually try to expose them to our own antigens to make sure they're not going to react. Now, there could be other things, though, other than just the breakdown of self-tolerance mechanisms. And those are triggers, right? Some of them could be environmental, like the question in the back from Stephen. Okay, take type 1 diabetes. Are there certain triggers that could actually stimulate this autoimmunity to flare up? If you know anybody with multiple sclerosis, okay, we have a close family friend that does, and I'm not going to share too many details, but there is a specific trigger in that individual's life that we all dwell on and know about. Okay? And for a lot of these autoimmunities, there can be a trigger point that takes place. Now, some of them can be environmental as well, right? Or they're genetic triggers. It's not like, oh, well, don't have funnel cake, for crying out loud. Okay? Maybe don't have 3 and then 2 things of cotton candy in one day. I mean, I just think that's a bad idea, even if your pancreas is, robust. Okay? But there could be genetic triggers as well that take place. As we age, we actually have DNA that's aging as well. And that DNA, as it ages, becomes less, the fidelity is less preserved. And so there could be genetic triggers that come in, and certain genes that are susceptible based upon inheritance causing a beginning of autoimmunity. So if you have a genetic susceptibility and there's failure of self-tolerant screening, then you're going to develop self-reactive lymphocytes, B and T lymphocytes. And that's how these things come about as they get into the tissue and cause these problems. Now, if we further take a look at some other specific examples, this is a type 3, systemic lupus erythematosus. And I want to show you this example because it's one that's pretty well described in the literature to have certain types of environmental triggers. Okay, and they happen to be sun exposure. So a lot of patients that wrestle with systemic lupus or even skin lupus, when they're exposed to large amounts of sunlight, a lot of times that's a trigger for them, and the lupus flares up. Okay, so systemic lupus erythematosus. There's really a handful of different types of lupus. Four main ones. The systemic one that we're talking about. There's a skin-only, known as cutaneous. There's a drug-induced lupus, and there's even one known as fetal lupus. Nuclear antigens tend to be the culprit, and the two initiators that we see are both genetic as well as environmental stressors. The environmental stressor is a lot of times UV radiation. Like, they had a really bad sunburn. They went to the lake, and it was a really bad sunburn, and then they started developing these symptoms of lupus. So lupus is a chronic multi-system inflammatory disease. UV light exasperates the symptoms. Okay, so patients tend to be a little bit more careful about staying out in sunlight. It's much more prevalent in women than it is in men, about 9 to 1. It's one of the more common autoimmune disorders. In some of our subpopulations, we see about an incident rate of 1 in 2,500 patients. All right. Yeah, 4 types. So systemic, skin, otherwise known as cutaneous, drug-induced, and fetal. Okay? So what's going on with systemic lupus? Well, it's all over the body. That's why we call it systemic. So this particular picture is showing some of the skin types of manifestations. It's this classic butterfly rash that comes over the bridge of the nose that you can see here in this young patient. Okay, I think I showed this well, maybe in the leg. Can you see it okay? That's a lot better. Okay, so this is not an adolescent, so you wouldn't expect there to be a lot of acne. This is part of this inflammatory skin response. This is called a malar rash, M-A-L-A-R. The other one is we get a lot of joint swelling. We get redness of the skin or skin erythema, like what you're seeing with this butterfly rash. We find vasculitis in various parts of the body. We see glomerulonephritis in the kidneys because we're developing, this is a type 3 hypersensitivity, we're developing antibodies that bind to antigens and they circulate throughout the body and they get lodged in certain places like skin, joints, blood vessels, the kidney. Also, there tends to be a central nervous system involvement. We don't really understand why, but we think that likely it's due to the vasculitis and the blood vessels that perfuse the brain. And so there could be some mental impairment or some sort of mental manifestations due to a reduction in blood flow based on the vasculitis that's perfusing the brain. So type 3. So another type example that I want to talk about is type 4, and this is rheumatoid arthritis. Now with rheumatoid arthritis, as another example in this last little segment, it's extremely common. It's extremely common. About 1% within our total population in the United States is affected with rheumatoid arthritis. A type 4 hypersensitivity because it involves helper T cells. Now, there is thought to be a B cell involvement in rheumatoid arthritis where there are antibodies being produced. And we actually think that the antibodies that are being produced may actually be in response to a collagen type 2. But because we know that helper T cells are involved, we call it and classify it as a type 4. Does that make sense? So what's going on here is we've got this erythrogenic agent we think is collagen type 2. And that's this antigen that's being seen. And then we stimulate with helper T cells the production of numerous cytokines. We activate B cells to form autoantibodies, and we call this sort of our rheumatoid factor, if you will. And then we have a whole host of different cytokines that cause an inflammation and a breakdown or a destruction of the joint. And so what's manifesting itself is this inflammatory disease within the joint. And if we look at what's happening with rheumatoid arthritis, we focus it on the joint, but we also see issues that show up at other locations like the skin, with rheumatoid nodules, and in blood vessels with obstructions. So it kind of starts in the joints, and it moves to other locations. The one that shows up in the joint that's very, very common in our population is we get this synovial destruction. So you can see on the far left this cross-sectional diagram of sort of a normal joint on the left and a rheumatoid joint on the right. And the collagen 2 that lines the joint is thought to be this arthrogenic agent, and that's what's actually stimulating this local inflammatory response with antibody production, but again, we call it type 4 because of the involvement of T cells. And we get infiltration of inflammatory cells and inflammatory mediators, and we get this pannus ingrowth of this inflammation. The joint swells under this inflammatory response, and we develop what we call ankylosis. Ankylosis is a word that refers to a bent joint or a crooked joint. It's from the Greek, which means bent. So if you look at the middle picture, you can see a low magnification of synovial hypertrophy. Synovial hypertrophy showing all of the swelling that's taking place, and you got the infiltration of the inflammatory mediators. On the far right, you're starting to get dense collection of aggregates. Right? These are lymphoid aggregates that are starting to form. These red arrows are pointing to these lymphoid aggregates, which are T cell bundles that are forming these lymphoid types of tissues within the joint itself. And of course, our classic pictures look something like this. So rheumatoid arthritis, it's going to target more than just the joints. It's going to target the joints, the skin, the vessels, at some level the heart. It can target the lungs as well as the muscles. You get this crooked or this bending of the joints. It's episodic, meaning it tends to flare up and then subside. It usually targets the small joints first and then moves on to larger joints. That's why the fingers and the toes tend to be more affected first. It's bilateral or symmetric, like we talked about, because you're talking about a systemic response as it's circulating around. It's not going to just target the right hip or the left hip. It'll eventually get to the hip, but it's probably in the hands first. Now, this rheumatoid arthritic factor has been identified in only about 80% of the cases. So that means 20% of the cases, we don't really know what the stimulus is. We don't know if there's an a rheumatoid arthritic factor, and we've only been able to identify like collagen type 2 being the culprit in about 80% of the cases. Now, the final slide of this particular lecture is this table. And this is a really nice summary table because it's not from your textbook. I pulled it out of a different textbook, but it helps to show you kind of the classifications of 1, 2, 3, and then 4. And you can see some of the features from a study tool that 1, 2, and 3 all involve immunoglobulins, right? And type 4 doesn't. You can see that usually type 1 is exogenous, right? Type 2 is something on the cell surface. Type 3 is soluble because it has to circulate in the bloodstream. And then type 4 targets tissues and organs. Okay? Transferred with antibody T cells. Okay? So this is a nice summary slide, in my opinion, that gives you kind of a global view in one slide of everything that we just talked about. Make sense? Helpful? Okay. Questions on this? That's thought to be the rheumatoid factor is type 2 collagen.